Okay, then we would like to welcome you to this co-hosted webinar by Qlucore and Nebion, where we are going to explore gene expression data from chronic liver disease. We are two presenters. My name is Ann Holmberg from Qlucore, and I'm doing training, support, and sales. And my colleague Hint from Nebion will present herself.
Hi everyone, it is also my pleasure to take part in this webinar. My name is Hind and I'm a scientific project manager at Nebion, mainly responsible for customer interactions and user support.
Yeah, and we will guide you through this webinar. And the agenda is that we will give you a little background for about 5 minutes, and then we will do a use case demonstration using both our softwares in one common workflow. And after that, we will have a Q&A session for about 5 minutes. So, and then please communicate with us through the chat. So our two softwares is Qlucore Omics Explorer from Qlucore and Genevestigator from Nebion. So let me just move this. And the challenge that many scientists face is that the size and complexity of the data sets really makes it hard for biologists to understand the data. And if you want to go out and compare your research, your data with published data, there are tremendous amount of published data, which makes it hard. But the combined use of our two softwares can help you with this challenge, which we will show in this webinar. So Qlucore Omics Explorer is an easy-to-use tool to visualize and analyze data sets and to highlight and find specifically relevant genes within your data set. And in Investigator, you can compare expression patterns from your data to thousands of transcriptomic studies. And with the two softwares together, you can explore public transcriptomic data sets with visualizations and enriched annotations. And in this use case that we will show today, we will explore chronic liver disease gene expression data. And we will use a data set on chronic liver disease that has been enriched with annotations from Genevestigator and has been exported from Genevestigator as a G-data file, which is Qlucore's internal data format. And then we import this data set in Qlucore, where we explore, visualize and analyze the data set with the annotations, and at the end we create a gene signature. And this gene signature we copy paste into Genevestigator, where we use it to identify similar data sets and do some other analysis. And when we have identified an interesting data set, we export this data set into Qlucore Omics Explorer and basically continue from the start again and start exploring this data set. So you can work with the two softwares in a continuous loop during your analysis. And now Hint will introduce the data set to you. So please have a go at that, Hint.
Thanks, Ann. So let's take a look at the dataset that we have at hand. We have chosen two studies from GEO, which use mouse models of NASH, with the titles that you see at the top of the slide. NASH stands for non-alcoholic steatohepatitis, which is a very common liver disease that is a progressive form of NAFLD, or non-alcoholic fatty liver disease. NASH is characterized by hepatic steatosis, inflammation, and fibrosis, and an optimal treatment has not been established yet. NASH can progress into end-stage liver disease and even into liver cancer, known as hepatocellular carcinoma. Therefore, research using translational animal models of NASH is necessary for better diagnosis and more effective treatment decisions. So in the two studies that we chose, The authors make use of two main mouse models of NASH without diabetes, which are established by feeding the mice a special high-fat and high-cholesterol diet, which we will refer to as CL, as opposed to the normal chow diet. And they generate their data using an Affymetrix gene chip DNA microarray analysis on liver tissue samples from mice with identical genetic backgrounds. In one study, this diet is also deficient in methionine and choline, and hence we will refer to it as MCD. Additionally, in both of the studies, a group of mice that are diseased, hence they receive the special diet, also receive a compound that is being investigated for the treatment of NASH. If we take, for instance, the MCD study, the compound that is being fed to the mice in addition to the diet is called metformin, which is an anti-diabetic drug for which the effects on inflammation and fibrosis were not yet defined. So if we look at the sample numbers in more details, we have 27 samples in total. In the first study, there are 5 mice that are fed the high-fat and high-cholesterol diet, 5 that are fed the high-fat and high-cholesterol diet together with a compound, and 5 that are fed a control diet. And in the other study, we have a very similar setting, but with 4 mice in each group, resulting in 9 in total for the control group. With that, I hand over back to Anne to start the demonstration.
Thank you very much, Hint, for this excellent introduction to the dataset. And now we will start the demonstration. So I will now open the dataset, and here it is in a GE data file that has been exported from Genevestigator. Here we are. And so in this case, the dataset was imported as a GE data file, but Qlucore also support a number of other data formats such as, or other types of data such as RNA-seq, single-cell, methylated DNA, proteomics, flow cytometry, and so on. And you can also download data directly from Gene Expression Omnibus and TCGA. So there are many data types that are supported by the software and many different formats that we can, data formats that we can import. Okay, but this specific data set we have now opened up in a PCA plot, and PCA stands for Principal Component Analysis. And this is an excellent way of showing your data in 3 dimensions. So we of course have many more dimensions in this data set because we are right now looking at 27 samples and 20,000 different genes. So we picked the 3 dimensions where we have the most information, and that allows us to visualize our data set, which is very, very useful. So when looking now at our 27 samples, because each blue sphere here in the plot is a sample, we see that the samples seem to divide up in 3 different groups. So let's try to find out what these groups are. So I go now here and look at the annotations. So these are annotations that have been curated and also some added by the Nebion team. So I will now pick the annotation diet formulation. And here we have the 3 diets, the normal diet, the high cholesterol, high fat diet, and the MCD diet. So let's color the samples by that. And then we see that there seemed to be a very clear difference between the 2 diets. So let's try to explore that. So let's take, so the pink diet here is the high cholesterol, high fat diet, and the yellow samples are the MCD diet, and the blue are the controls. So let's take away the controls for the moment, and let's focus on the 2 diets. So now we will do a statistical test in order to find genes that are good at separating these 2 diets. So we do a t-test, which we call a two-group comparison on the annotation diet formulation, and then we pick one of the groups. And then we just drag the statistical slider here in order to filter down to the best genes. So I filter down to a Q value of 0.01. The Q value is the same as the false discovery rate or the corrected p-value. And this is what we should focus on. As you know, we should not focus on the p-value. And then we can also add a fold change filter when we do a t-test. So maybe I would like to filter to a fold change of 2. And now I find 2,200 genes. So I would like a little shorter list. So I continue to filter down to something like that, 1,600 genes or so. Okay, and we can see that these genes, if we just look at the samples, we can see that these genes are really, really good at separating the two groups from each other. And if I would like to have a closer look at these genes, I can open up a new synchronized plot and make that a heat map like this. And let's press auto here so that we see everything in the heat map, and also let's hierarchically cluster the heat map like this. And let's color the heat map according to diet formulation so we see what is what. And now we can clearly see that, so I will just close the chat for the moment like that. So now we can clearly see that there is a very clear difference between the two groups. We have here upregulated genes for the high cholesterol, high fat diet, and here we have upregulated genes for the MCD diet. And if I zoom in a little bit here, we will see the gene names like that, and I can just scroll down like this and have a closer look. And when I want to see everything again, I just press auto. We can tile the plots and have a look at them side by side. And if I now would like to save the list of genes that I've found, I go to the Variables tab. I make a copy of the active list. I give it a name, so genes separating diets. And down here I see the content of the list, and here I would like to add more information. I would like to have the gene symbol, I would like to have the p-value, the q value, and maybe also the fold change. Now I have all this information in the list. Then I can sort on any of the columns, so they are now sorted according to q value, and I have the best genes on top here. And this can easily be exported to a TXT file, so it's very easy to get this file as this information as a text file. And now maybe I would like to have a closer look at some of my best genes. So let's open up a new synchronized plot and make that a box plot. So I now change this to a Boxplot box, and I need to define the axis. So I would like to have diet formulation on the x-axis, and then I would like to select with the mouse which genes to show in my list. So I picked the 4 first best ones. Now I have them here. And let's see that I want to have gene symbol on the y-axis. So now I have my 4 best genes here, and you can see that they are 3 of them are upregulated for high cholesterol, high fat, and 1 of them is upregulated for MCD. And if I have a closer look at one of them, we can, for instance, see the individual samples. We can also decide which p-value we would like to show up here to show the separation of the groups. So we have 3 different p-values to choose from. Then I can tile the plots, and all of these plots can be very, very easily exported in many different file formats by just doing File Export Image here. Like that. Okay, and now we will focus on another annotation. So let's reset this analysis back to 0 and and then also put back all the samples like that. So now we will have a look at the annotation stimulus, and let's color by that as well. So now we can see that within the MCD group we have both untreated samples as well as samples or mice that have been treated with the compound, and then we have the normal controls. And in the high cholesterol, high fat group, we have untreated samples, which are the red ones, and the green ones are treated with the compound, and the white are the normal controls. So now I will focus on the MCD diet here. So I take away the other samples for now, which I can do on the fly, as you see. And now I would like to do an F-test here, which we call a multi-group comparison on the annotation stimulus. So then again, I just drag the statistical slider to maybe some more genes. So I'm now just looking at how many genes, and I want around 1500 or so. There we are. And then we pull up the heat map, which is actually already open, and that automatically updates. So that happens on the fly. And we will color this heat map also according to stimulus, like that. So here we have them. So let's just make this a little smaller. And what we can see here is that this is the treated samples here, the pink ones. And we can see that there is a tendency for some of the genes here to become less red and more blue. So the treatment means that these subjects are becoming more normal. And we see the same tendency here on a couple of genes. And you can maybe see that also in the PCA plot, that the pink group goes towards the blue group a little bit. It's not a very strong signal, but still you see the tendency. And now I would like to create a gene signature. So I want to compare the MCD untreated group with the normal controls. Then I take away the pink group for the moment. And now I will open up a volcano plot. We can have a look at our genes there. So let's just do a t-test and change the statistics here. And then we will change this to a volcano plot where we can see our genes. So here we have them. And let's color these genes on fold chains, for instance. So here we see them. And if I want to label some of the best genes here, because I have the best genes up here, as you know, just circle like this, or click on an individual gene. I would like to label that one and that one, and I would like the label to be gene symbol. And then when creating the signature now that I want to send to the Investigator, I want fewer genes. So I want around 50 genes or so. So I filter to a fold change of 2, and then I continue to filter on the P and Q values down to say 57 genes. That's fine. And I see them here, and I also see them here because the heat map was also updated at the same time. So now I go to the variables tab and I make a copy of this list and call it MCD versus normal, like that. And then I right click and copy differences like this. Differences means the log2 ratio. And then I move over to Genevestigator, so another software that I have open on my machine here. Open the signature tool and go here, and then I can just enter the signature by doing paste. So now it's in Genevestigator, and now Hint will show you what you can do with this gene list here. Now I will just make you the presenter hint.
Yeah, please.
And you can take over. Yes.
Yeah, thank you, Anne, for handing over and for creating this exciting signature. Let me just share my screen. Perfect. Okay. So before I move on with the demonstration in Genevestigator, I will just give a brief introduction about our platform for those in the audience who are new to it. So we make gene expression datasets from public repositories available for compendium-wide analysis. Here you can see an overview of our pipeline. We download raw data from public repositories such as GEO and Array Express. The data then runs through an in-house quality control normalization process before our curators add extensive annotations according to set ontologies that enrich the data. The studies are then integrated into our database, where using our tools, this data can be queried and compared. So one can use Genevestigator to compare your own datasets and put them into context. You can also profile your targets across tissues and diseases. You can also find co-expressed genes or specifically expressed genes. So in this use case, we identified genes that differ in expression between MCD-fed and control-fed diet mice. And I would like to pose two main questions that we would like to answer today in this Genevestigator Qlucore combined demonstration. The first is, can we verify whether the signature that Anne has created is really specific for the disease NASH? And secondly, can we validate disease biomarkers? We will answer the first question by choosing a dataset that includes other published studies, which we can use to compare to our results and to look for other similar findings for verification. So let's get started. I have opened up Genevestigator, as you can see here, where we will paste the signature that Anne has created using Omics Explorer. Firstly, I have defined a selection of datasets that you can see up here on the left, which include all experiments from mouse that we have on the Affymetrix platform, which is the largest resource for the microarray platform. If I click here on the triangle, you will see that this includes many different datasets with a total of over 12,000 samples. In the large window on the right, you have an overview of all the available tools. The compendium-wide tools allow you to analyze multiple experiments at the same time. If you are interested in looking at one gene or at multiple genes, you can load it down here on the left. Since we are interested in the signature that we have received and we would like to visualize the genes in the selected datasets, we can directly go to the signature tool in the similarity search tools down here, as Anne has previously shown you, and choose to enter our signature. I will just copy paste the signature that I have received from Anne and give it the name MCD versus normal, and choose the right scaling of the data, which is log2, equivalent to differences in Qlucore, and select that my data is relative, since it's a experimental versus control. Then I will press OK, and we are ready to start the analysis. You have to accept that these are the genes that you have defined, and now the signature will appear up here. As you can see, there are some genes that are red, and these are the upregulated genes, and there are also green ones which are downregulated. So we have a nice mixture of both. We have set the tool to the most similar, and what the tool will does is it will search for the most similar profiles as possible. We will change the distance measure to a Pearson correlation coefficient and then click Run, and this will display the results. So these results are based on the values of expression, and for every row here, you are looking at a comparison between two groups of samples, which we call perturbations, and every column that you see here is one of the 57 genes that we have selected. As you can see, many of the comparisons that are defined are comparisons between steatosis or liver fibrosis. There are also a few non-alcoholic steatohepatitis studies, And there are even some comparisons, such as the one down here, between fat diet study and a low-fat chow fed liver tissue. And you can always get more information about each row by placing your tooltip on top of that study row. Since we see so many NASH-related dietary models, it means that we could identify several comparison sets that give a similar response to the dataset that we explored in Qlucore's Omics Explorer. This represents one way in which we can verify results, since here we were able to identify other studies than our own dataset in which we can identify similar patterns of differential expression for this set of genes. Now, if we look at more details in the list, we can identify several comparison sets that have similar profiles, but which represent other diseases, such as a liver aging study, like the one up here. There are also certain drug treatments, such as the sodium arsenite study here, which, if you look at more detailed information, is actually performed in lung tissue, not in liver. And there are also some involving a liver cancer, such as the one you see here, which is a hepatocellular carcinoma. And as you might remember from the introduction, hepatocellular carcinoma is a possible outcome of NASH. So we will record the number of the study, MM00465, and come back to it at the end of our demonstration. As you can see, this approach helps you to identify disease expression patterns, and it reveals the true value of conducting a compendium-wide analysis. It demonstrates that combining data from several organs, several diseases, and several cell types can bring you a lot of information if you start from a single signature. Now, as you might recall from the introduction, in the original study design, the experimenters also treated mice that had been fed the MCD diet with a compound, metformin, which they wanted to investigate as a possible therapy for NASH. Since the gene list that we have been using so far was defined based on a comparison between diseased and control, we might be wondering how the samples that are treated with the compound are affected in terms of expression of the selected gene list. In order to structure comparisons that we are looking at and to find clusters of similar genes and samples, we can enter the hierarchical clustering tool, which you see up here. So we will stay on the same gene list of the ones that we previously defined, but we would now like to add a new dataset where only our one study of interest is included. For that, I can go up here to add new data selection and search by keyword, and I will type in the number of the study, which I already know, 00463, search for it, and I will give it the name. So this is the original MCD dataset that we used for defining the signature. As you can see, this now appears here with only one study and 12 samples in total. We can now choose the samples from the tab so that we can see on every row one of our 12 samples, and then we can cluster by genes and conditions according to the Pearson correlation. If I now click Run, you will see a demonstration that is similar to what Anne has shown you when she performed the Heatmap analysis in Qlucore's Omics Explorer. What we can see here is that the 4 biological replicates for every condition cluster closely together. And these you can see here using the nodes. And that the expression of our 58 genes seems to be most different between this control group as compared to the 2 diet-fed mice. Now, one question that we could ask, which we originally posed, was, can we identify and validate biomarkers of this disease? So in this plot, we can identify, for instance, 2 genes that are strongly differentially expressed between the disease and the control, which would allow for a better visualization of the compactness of the clusters. So I will choose 2 genes that show a strong and different expression between the sample groups. For that, I will use the UBD, which you see up here, and MBL2, which you see here on the right. So why I choose these 2, I will show you in a second. So if we deselect all the other genes and keep only these 2 that we just chose, this would be the MBL2 and the UBD, you can see that the expression is intermediate in the metformin treated group, and it's either very high or very low in the two other groups. So if we go to the 2-gene plot tool and stay with our selection of only 2 genes and 12 samples, we can color according to the stimulus this scatter plot. Let's give the MCD group red and green to the metformin treated group and blue for the controls. And what you can see here is that the samples form very nice clusters that separate very clearly, and that those MCD metformin treated seem to move away from the diseased towards the control. And these results actually verify what the original paper had shown, that the metformin treatment seems to ameliorate the diet-induced inflammation and fibrosis. So this is one way in which we can choose biomarkers of the disease that can be verified, and this graph can easily be exported up here under results export image. So as you can see, there's a lot more analysis that one can still do. And what we would like to end with is to bring one of the studies that we identified in the signature tool back into Qlucore for further detailed statistical analysis. So let's take the example that we identified before with the hepatocellular carcinoma study. As you remember, we recorded this number. We can now go back to the data selection and add this experiment. Like this. And I will give this one the name of hepatocellular carcinoma study. This will appear here with, again, only one study with 39 samples. So if we briefly want to look at what kind of samples this study includes, we can go to the Home button and go to the condition search tool, which we call perturbations. And now you can see that every row represents a comparison. And if I display it using a heat map tree, this will show me also in which part of the tree this belongs to. So we have a few comparisons between a fatty liver disease knockout mouse models, and we also have a few liver neoplasms, hepatocellular carcinoma. And you can see that for the 2 genes that we selected, we can see the expression, and we can always reselect all 58 genes to display this like this. So now to explore this data set further in detail, we can export this single experiment from Genevestigator and import it into Qlucore. So for exporting, I will go up here to Dataset, Current Selection, Qlucore, and this will export it as a GE data file, which is the data format that Qlucore uses. This export is available for special Genevestigator users, just as a side note. So since this takes a few seconds, I've already exported this data set, and I can double click this, and the file will open in Qlucore's Omics Explorer, as you can see here. So we are now looking at Qlucore's Omics Explorer, where we can easily visualize the annotations that my team at Nebion has added. So for instance, we can color the samples according to neoplasm. As you can see here, we have the 4 samples that have the liver cancer. Maybe more informative would be to color according to the stimulus. So here you can see the multiple sample groups according to the annotation that is attached to it. And you can keep exploring your data sets as we have shown you in the beginning of this webinar. Okay, so to summarize, in this webinar, we have explored 2 datasets on chronic liver disease, starting with the data that has been enriched with annotations from Genevestigator. We explored the dataset and the annotations in Qlucore's Omics Explorer, where Anne examined 2 different dietary NASH mouse models and used the differences between the control-fed mice and those treated with the MCD diet to create a gene signature. We then imported the signature into Genevestigator, where we compared it to a whole compendium of other datasets, and this allowed us to identify similar datasets for further exploration. We additionally identified biomarkers of NASH in Genevestigator. And as a last step, we exported this single dataset that we identified to be interesting for further analysis in Qlucore's Omics Explorer. So by combining Genevestigator and Qlucore's Omics Explorer, you get an end-to-end solution for gene expression analysis. These complementary tools provide you with a synergy of analysis options for your data, combining a deeply curated database with an interactive statistical toolset for visual exploration. And what we have shown you today is just one example from a particular workflow. But please go ahead and explore yourself. Qlucore and Genevestigator can be used in combination to strengthen your particular research question. And if you are interested in trying out either of our two softwares, you can download free trials from the websites listed on this slide. I would like to thank you for joining us today, and we are now